Hey, Andrew, whenever you're ready, we can let people keep joining in, but we can get going here in a second.
Jacob, is that shared correctly?
Looks great.
Okay, do I need to do anything else?
No, I think whenever you're ready, we're recording and you're ready to get started whenever you like.
We're recording live? Jacob?
Yes. Yes,
We're recording. Okay,
we're good to
great.
go.
Okay, good day, everybody. Thank you for joining. I'd like to welcome you to the Department of Energy Isotope Program virtual seminar series. Today, we'll be discussing the radioisotopes of Lead-203 and Lead-212. Last year in the 2022 Lead-203, Lead-212 user group meeting, which was hosted by the Department of Energy Isotope Program, the scope of the meeting was increased to include that of Lead-203, and we're glad to continue this trend this year. My name is Andrew Burgoyne. I will be moderating today's session. I'm a radiochemist at Oak Ridge National Lab. I'd like to thank the Department of Energy Isotope Program and the National Isotope Development Center, the NIDC, for making this virtual seminar possible. I'd also like to thank Jacob Haskew at the NIDC for all the work that he does in the back end, behind the scenes, which has made this virtual seminar series happen. I'd like to share with you who our speakers are today. We'll have 6 speakers. I will give a brief introduction into radium-224 and lead-212 generator production here at Oak Ridge National Lab. Second, we'll hear from Dr. Matt O'Hara from Pacific Northwest National Lab. Following this, we'll get into the application side of our presentations. We'll hear from Dr. David Bauer from Memorial Sloan Kettering's Cancer Center. Then we'll go back to the isotope production side of things to hear from Dr. Jennifer Bartels at the University of Alabama, Birmingham, to talk on Lead-203. Then on to Dr. Kurt Zinn, Michigan State University. And last but definitely not least, we'll be glad to hear from Dr. Michael Schultz from Perspective Therapeutics. Just a bit of housekeeping. After all the talks, we'll have a moderated question and answer session where any questions you may have can be raised to the panelists. You should find a Q&A tab at the bottom of the meeting screen. Since we're going to be answering the questions after the fact, if you have a particular question, please identify to whom your question is addressed. In addition to this, I should note that if you have any business-related questions that involve some NIDC-related topics, things like quotations, generator or radioisotope pricing, distribution or production schedules, if you could please go directly to the NIDC with these questions. You can find their contact information as well as a host of other information at isotopes.gov. We'll now get into the presentations, and I will talk about radium-224 lead-212 generator production at Oak Ridge. So here at Oak Ridge National Lab, we have a history that's rooted in isotope production, enrichment, and distribution. It began in the 40s, thanks to the construction of the graphite reactor and all the enrichment technologies that were developed in the early days of the Manhattan Project as part of the war effort. In 1946, the first shipment of a reactor-produced radioisotope for medical applications, carbon-14, was produced at the graphite reactor and was shipped out. Until 1963, thousands of shipments from a variety of 60 different isotopes were used for cancer treatment and as traces for academic, industrial, and agricultural research. In the late 40s, 2 geneticists, Leanne and Bill Russell, started a mouse genetics project to study radiation's genetic effects on mammals. And based on their studies, specific recommendations to avoid diagnostic X-rays in human pregnancies were adopted worldwide thanks to them. In the early 90s, Russ Knapp, a legend here at Oak Ridge, developed the tungsten-188 radium-188 generator. Radium-188 can be used to treat cancer-induced bone pain and arthritis, prevent the development of smooth cells and coronary arteries. In 1997, Oak Ridge began producing actinium-225 and is one of the leading producers, and it's used in targeted alpha therapy. In the late 90s, the DOE launched a research project that studied the effects of very low dose exposures. Research was conducted at Oak Ridge and 2 other national labs to understand radiation-related health impacts. And since 2017, Oak Ridge began producing actinium-227, which decays into radium-223, which is the primary ingredient in the only alpha-emitting approved radiopharmaceutical, Xofigo. So back to lead-212. Lead-212 is a half-life, 10.6 hours, and it's usually isolated from one of its longer-lived parents, radium-224, which is a half-life of just under 4 days. I'll show you on the left here some of the decay chains. You see where radium-224 sits in that chain and how it's related to lead-212. Radium-224 itself is a decay daughter of thorium-228, which is half-life of just under 2 years. And thorium-228 can be obtained from a variety of different ways. It's a decay daughter of thorium-232, and if one pursues that particular production route, then it would require large masses of significantly aged thorium-232 to be processed to obtain significant amounts or usable amounts of thorium-228. It's also a decay daughter of uranium-232, which is a much longer half-life, and this can be milked annually or biannually for thorium-228. And that's one of the routes that in the past we've used here at Oak Ridge in building 4501 to supplement our thorium-228 count. But our primary source that we're using for thorium-228 is from the production route, as I've shown you with the red arrow, starting from radium-226, and that thorium-228 can be co-produced by double neutron capture to produce thorium-228 in the production of thorium-227, actinium-227, sorry for that. Oh, we're not advancing. So generator production at Oak Ridge was established in 2014, and we're currently separating that out from our stock of thorium-228 using an anion exchange column and then loading that purified radium-224 onto a cation exchange resin. The generators that we're producing have an activity of up to 20 millicuries and can be milked periodically for lead-212 or bismuth-212, depending on the uses of the studies. And these can be used in targeted alpha therapy. We also supply thorium-228 at Oak Ridge National Lab, and this can be purchased through the ANDC. I'll talk about that later. So the generators that we're producing, you can see that in the center diagram, you have your loaded generator resin. So this cation exchange resin is contacted with radium-224, and this has an inlet lead, an outlet lead, and sits in an 1.25 inch the generator pig. And the photo on the right really shows the Cherenkov radiation that comes from the radium-224 on this particular MP50 cation exchange resin. You can see above and below the purple or light lilac part of the column, you can see an orange plug below and beneath, where we're not contacting with that MP50 with radium-224. So the low glow of the alpha particles coming off of the radium-224. So this particular generator has been stripped of lead and all of its daughters. You can see the Cherenkov glow coming from that resin. And that's how we're shipping that out. So looking at the dilution profile, we know that we can separate out lead-212, and this is we're generally doing with 2-molar hydrochloric acid. If one's interested in isolating the bismuth-212, you can do that exclusively by following the instructions that we provide with it. And here I've got a little image, a little video clip showing as we do a separation of lead-212 and its daughters from radium-224. You can really see the stark contrast of the Cherenkov radiation of the daughters as they're stripped, as they're eluted off of the resin, and the remaining radium-224 that remains behind the weak Cherenkov that's visible from it. Let's get to the next slide. So in the near future here at the lab, we're currently have our glove box shield that's to be installed. It's now on site. We're happily awaiting its installation. We plan to use this to minimize the worker dose as we are doing the chemistry to purify radium-224 from thorium cow, and as we produce our generators. And this would, of course, also allow us to increase the radium-224 loading activity of our generators or increase the frequency at which we're producing generators if this demand exists from the users or customers of our generators. So in terms of ordering or availability, mentioned that one can purchase radium-224 generators. You go to the isotope.gov and navigate to your catalog, and you will see a variety of things. You can also purchase radium-224 as a chloride solution or as a nitrate salt, if you wish to fabricate your own generators. And thorium-228, as I mentioned, is also available to purchase through the NIDC. And that's all I have to share. And thank you very much for your attention. We'll now proceed on and stop sharing. Matt, would you like to share?
Sure.
Thank you.
Okay, is it up? I guess so.
Looks
Okay.
good.
Yeah, looks good.
Thank you, Andrew. I'm Matthew O'Hara, and I would like to acknowledge Lucas Moran Brenner, who is essential in this work. Okay, so several years ago, PNNL set out to develop this generator production process as a fully automated fluidic system that would purify radium from thorium-228 feedstocks on the front end, and on the back end, take that purified radium, homogeneously mix it onto cation, exchange resin, form it into a slurry, send it downstream to pack the column, all automatically. And so we've been developing this. It's been quite an adventure for several years now. Initially, we were developing a chemical separation process to make this happen with getting the inline radium purification process, and develop the slurry handling system on the back end. When we had that pieced together in FY 2021, we set the prototype system up in hot cell laboratory within our facility, and started testing the system at low micro and millicurie levels. And then in FY 22, we were able to scale up to clinically relevant levels. And in this most recent work, we continued those operations and also evaluated the process and the performance of those generators up to 30 millicurie levels. And so the motivation to all of this was to take this technology and transition it from research and development to a level that we could use it for routine production. And most importantly in all this process is that it would minimize the radiological dose to the staff members that are responsible for supplying these generators for customers into the future. So just a high-level overview of how we approached this problem. We basically split the process down into 3 individual fluidic modules, which were later interlinked into one continuous chain. The first module is the radiochemistry module, where we obtain high purity radium from thorium feedstock. And we also separate out the high dose progeny from the radium during that process. And then very importantly, this radiochemical module has to be capable of recycling the valuable thorium feedstock so it can be reused for future generators. Module 2 is a simple radium preparation step just to get that radium into an ideal chemical form for optimal resin absorption. And then module 3 is where all the slurry handling and column packing operation occurs. So when we had these individual modules developed, we chained them together, moved them into the hot cell facility that you can see the pumpkin orange box in the background. That's where the thorium-228 feedstock is kept, and there's an umbilical that connects this temporary glove bag system for evaluating the prototype to the hot cell. So we have an interlink between the two. The thorium feedstock can be pulled from the hot cell into the fluidic module, do what it has to do, and at the end recycle that thorium and push it back into the hot cell. And so we've spent quite a bit of time optimizing the process efficiency of this system, and so we've whittled this process down to about 2 hours of duration to get column packed, including a pause while we await quality assurance testing. And so about 2 hours from when we originally accessed the thorium-228 feedstock in the hot cell to actually having the generator delivered into a shipping container. And human intervention in this whole process is limited to the thorium stock insertion on the front end, which is basically a zero dose operation, because that is handled by manipulators in the hot cell. Out the back end of the process, when the generator is prepared, there is human interaction to go in and disconnect the generator from the inlet and outlet feed lines that attach it to the fluidic system, and then of course surveying the generator out of the radiological enclosure and getting it loaded into a drum. So we have been tracking the performance of this fluidic system over the 13 high activity generators that we've prepared over with our R&D testing at this point. And so one thing of note is, over many orders of magnitude, we see no differences in the generator preparation performance whatsoever, and that the radium yield onto the generator, so the fraction of radium that's contained in the thorium feedstock at secular equilibrium, is very reproducible at 84 plus or minus 2%. And that the thorium feedstock recycle is essentially quantitative. And so we have virtually no thorium ending up in our waste streams, basically. The other metric that we've tracked is how much thorium-228 actually makes it through all three modules and ends up on the generator column. And so we're seeing decontamination factors of over 10 to the 7. So basically, for example, here I put on a 15 millicurie radium lead generator, it would be expected to contain a few hundred picocuries of thorium when everything else decayed away. So concurrently to this last year of the R&D based evaluation of the system and the generator process, we began transitioning to production. So we initiated efforts to become an official lead-212 generator supplier for the NIDC. And so in this winter, we will begin producing for them to fill customer orders on the 10 to 15 millicurie range. So in the effort to get into production status, there's been a tremendous number of activities that we've been involved in, including laboratory renovation, having a proper hard-walled glove box designed and procured. We're awaiting delivery for that now. Adaptations to the hot cell, so you can have connection. We can have basically hard connection between hot cell and the 3-module glove box system. Designing customized type A packaging for customers that can accept either yellow 2 or yellow 3 shipments, et cetera, et cetera. So when we prepare the generators in this laboratory, we've also been evaluating the generator performance for each one that we prepare. And so we basically perform milkings at 24-hour intervals across 10 to 12 days for each generator. And so we've evaluated different approaches to milking the generators, where we have three milking approaches, and we basically cycle through day 1, 2, 3, and then back to the method 1, 2, 3, etc. And so we look at a few things. First is the lead yield in your milked product fraction versus the other fractions that you collect into that, and I'll report on that. And also the radionuclidic purity of the milked lead product. So traditional milking is basically the 2 molar hydrochloric acid, where we deliver 1 milliliter of milking solution followed by 1 milliliter of water. And in that most traditional approach to milking, we see here across, well, this is a population set of 37 milkings across all the generators that we've evaluated, so about 94% in that milk product fraction. The other approach that we've evaluated is to insert a catch column at the outlet of the generator, and this is basically a 100 microliter resin bed of MP50 resin, which is the same resin that is used in the generator itself, just as an extra preventative to assure or to minimize any breakthrough of radium from the column. In this case, since there is additional volume that we're putting in line, our lead product yield does take a little bit of a hit, but as you'll see in a future slide, the radionuclidic purity goes up. And then the tandem column approach. So this is a published approach where the lead is milked with the HCl solution as normal, but is passed through a lead resin column where the lead is bound to the secondary column and then is taken through various washing processes and is ultimately eluted from that secondary column as in a buffered acetate solution. And so so we call this the tandem column approach because of this secondary step in there. And here we get very high yield in the product fraction, similar to the traditional milking process. Another aspect that we look at is the radionuclidic purity. Really, from a radionuclidic standpoint, what that really means, since the amount of thorium-228 on this resin is so incredibly low, we're really looking at any trace amount of radium breakthrough from the column as we go through the 24-hour milking events. So we basically we milk the generator, and then we evaluate it on dose calibrator over several days, and you can see on the screen here an example of 2-component exponential decay that we model with sigma plots, and from that we're able to determine the radium breakthrough fraction from that. And so we do this across all the generators, across the 3 different milking methods that I've just presented. And so these are the findings that we found. So for the traditional, with the catch column, as you see, we go down in the amount of radium, so that 100 microliter additional supplemental catch column does work to drop the amount of radium in the lead product stream, and also the tandem column approach does very well. And that's my last slide. So I would like to acknowledge the DOE Isotope program for funding the R&D into this work and for supporting us in the move towards production for NIDC. Many people are definitely involved in this process that I would like to thank as well, and I will stop there.
Thank you, Matt. David, you have some slides you can share with us?
All righty.
Thank you.
Perfect. Good. First things first, Jacob, thank you so much for organizing all of that. I know it's a lot of work and we really appreciate that. And thank you all for joining this meeting. So maybe you're already thinking about targeted alpha therapy for a while, and you're thinking about starting with Lead-212 or doing more experiments with Lead-212, and all you needed was a little push. I hope this presentation is going to be your little push for that. We're talking about preclinical perspectives. So we have two nuclides here. That's Lead-203, which is a really fantastic SPECT imaging nuclide. You have two window settings for SPECT with Lead-203. That would be the X-ray setting, basically technetium, and you also have a mid-energy window 280. You can do both. In the little image down there, you see a mouse I imaged 2 years ago that was injected with free lead. Free lead usually goes to the liver, spleen, kidneys, and you see a little bit of joint uptake as well. So SPECT with Lead-203 is fantastic. On the other side, you have the therapeutic nuclide, Lead-212. And if you now think about a clinical perspective, sometimes it's really hard to get both nuclides to the same location at the same time. So there was a lot of endeavors going on to make SPECT imaging with Lead-212 also possible. And indeed, at least planar imaging with a limited amount, let's see a few millibecquerels per patient, gives already quite usable and quantitative SPECT images, even with Lead-212. So both of them can be used for imaging and will bring the advances to the clinic, hopefully very soon. When we talk more about Lead-212, you see that the rays that are emitted are not only coming from Lead-212 itself, but also from the daughters, which when you actually use that nuclide for imaging, it might give you a little bit more blurry images because all of 20% of the emission is coming from the daughters, at least when we're talking about the X-ray range. Now, one word of caution as well, when you use Lead-203 for your studies, Lead-203 perfectly mimics, of course, Lead-212. But Lead-212 itself is not an alpha emitter. The daughters are. So first of all, you need to prove that the daughters, bismuth and polonium, decay in situ with Lead-212. But as you will see later on, this is no problem. In most of the cases, this actually happens. Good. With this being said, if you start your first clinical studies, I would say you would like to start with Lead-203. It has a long half-life. It gives you a lot of freedom. If you start with Lead-212, you want to be sure that all of your studies and analysis are done within 2 days. On the other side, when you think about the clinic, Lead-212 is just ideal. After 2 days, basically, therapy is done and the nuclide is decayed, which gives you perfect control about toxicity, and that invites you basically to do fractionated dosing. And that's where Lead-212 is really going to hit the clinic. Fractionated dosing, I think, is coming up. Talking about the chemistry of lead. So lead is a very fascinating nuclide when you think about its chemistry. Just before lead, the lanthanoids are squeezing in, and this changes the chemistry completely. The core is quite heavy, and the electrons need to be quite speedy to not collapse into the core. Now, quantum mechanics kick in, the electrons become quite heavy as well. And what happens now is that the 6s orbital really shrinks. It shrinks a bit into the core, and that's why lead is rather Lead-2+. Only the p electrons like to interact, and the s electrons to shield it. This gives us now some challenges, because this s orbital that is not interacting normally is a lone pair in the nuclide. And this can partially shield lead, which means if you have coordination numbers lower than 6 and a chelator that is not very strongly interacting with lead, this lone pair shields from one side. On the other side, if you have a very strong chelator and coordination number above 8, you can actually dislocate this lone pair and have a whole directed chelation sphere. This is very important if you think about radiopharmaceuticals, because what happens if you have this lone pair is the lone pair can interact with other molecules like water, hydroxychlorides. It will weaken the overall complex because most of the time this interaction happens over p star and sigma star orbitals, but it also can change the charge and the behavior of your radiopharmaceutical. And what also can happen with this lone pair, it might interact with proteins and can also cause what you often see, the stickiness in kidneys and liver. So there's still a lot to do actually creating better chelators, especially for Lead-212. Which chelators can we use so far? Okay, all cyclin-based chelators are wonderful for lead. Here we have DOTA and TCMC. When you look for a perfect stability constant, it would be around 20. So you might think DOTA is just the perfect chelator for lead. That is true, but for lead, we don't need the most stable chelator. We need a good enough chelator because we only have to deal with a half-life of 10 hours, which means TCMC is, and that's why it is the standard one, still a pretty good chelator, and it has one big advantage. It's very lead specific, which means if you're just starting with your studies and you might not have the highest purities of everything, TCMC will always do the job. On the other side, DOTA might be more sensitive to impurities, but it also labels all the other nuclides, actinium, yttrium, lutetium, and so on. If you look at EDTA and DTPA, they're a little bit less stable. These are the perfect ligands for stripping or H+C ITLC. Closer look to TCMC. How does it coordinate? Surprisingly, it does not coordinate how many people draw it in the literature. You have a tautoremism going on here, and it coordinates over the softened oxygens and over the nitrogens in the cycling. If you now look at the crystal structure, you can see what I talked about before. You have this gap caused by the lone pair. So even though TCMC and DOTA are really good chelators for lead, they do not really cause a perfect homodirected chelation. They actually only do this partial chelation. So there is room for improvement. Now, thinking about translation, we have this spiral of theragnostics, and we talked already that we want to use Lead-212 and Lead-203. We know the chelators, linker, that's our own lecture for itself. Which vectors can we use for lead? Which vectors are feasible? With a short half-life, maybe not jump into antibodies directly, but small molecules and peptides are just perfectly suitable. And when you look into first clinical trials, we of course have the standard criminals, PSMA, somatostatin. We have some chemokine, gastrin-releasing peptide receptor, and melanocortin receptors, and even a study with HER2 and trastuzumab. What all of these receptors and vectors have in common is, upon the binding, they quickly internalize, relatively speaking, of course, which is extra beneficial. Because when you internalize something, you don't really need to worry about the daughters, especially bismuth is of concern with a 60-minute half-life. But once in the cell, it actually is captured there. We proved that, and that is very nice. There's also a downside to fast internalizing vectors, and that in most of the cases, these are getting chopped up quite quickly in endosomes and getting excreted from the cell again. So there's a big downside if you would match these with actinium, because only 60% of actinium actually does a job in the cell. The rest of it would get excreted again without doing any damage to the tumor, maximally only doing damage to kidneys. So lead here with a half-life of 10.6 hours is just perfect for these quick internalizing targets. Now, this wouldn't be a radiochemist presentation without talking at least briefly about radiolabeling. And I put a lot of literature on that slide as well, because there's a lot of citations already going on. Lead is not a newcoming nuclide. You can read about it. Here, just the highlights. When you use lead, you want to reconstitute your precursor in a buffer at a pH around 4.5 to 5.5. For most chelators, this will do the trick. You add your radioled, and now think about if you're using older lead or you have some doubts, repurify your lead using the lead resin. This was introduced previously as the dual tandem column approach. After that, really check your pH again. If it's too low, you might just protonate your chelator. If it's too high, lead and bismuth actually might form hydroxides and oxide species, and this will definitely interfere with your radiolabeling. Incubation time is no problem, normally at room temperature with TCMC after 5 minutes done, with DOTA after 10 minutes, most of it is chelated. Now comes the tricky part for your injection. Since you may have purified your lead or you will purify your radiotracer, your daughters are not in equilibrium anymore. So now you cannot just put it in the dose calibrator like you would do with lutetium. You actually have to either wait 6 to 8 hours before it's reaching equilibrium again, or you go directly for the mission of Lead-212, calibrate your germanium detector, your gamma counter, and do a separate measurement only to estimate basically your activity. It's an extra step, but it will not stop you from doing it. And then at the end, the QC. HPLC is mandatory, but I would also suggest, if you can, do scintillation detection as well, because then you can basically co-evaluate chelated lead and chelated bismuth, which is an alpha emitter and has no big gamma emission. So we talked about small molecules and peptides as being most feasible for Lead-212. In the loose lab, we do a lot of pre-targeting, saying we also want to use antibodies. With this concept that we are using here, we inject the antibodies in our subjects, mice, and let the antibody circulate till it's in the target, and the rest is cleared. Our antibodies are modified with transcyclooctene, TCO, and when this TCO finds its counterpart, the tetracene, they click and they create an irreversible bound. So which means after the antibodies accumulated in the tumor, we would inject our tetracene modified chelator. In this study, I used DOE3A, and this will quickly accumulate in the tumor, and the rest will quickly be cleared renally. You can see that here in our diagram, but pictures are even better. So we have some SPECT images with Lead-203 attached down here. You see that you have quick uptake after a few minutes to hours in the tumor. The excessive radiotracer finds its way to the bladder. And over the next few hours, you have constant great uptake in the tumor. So pre-targeting is really another alternative to deliver lead. So we did this approach with Lead-203 and continued that with Lead-212 to show first therapy results. As you can see here, we have 3 cohorts, the 30 microcurie, 60 and 100 microcurie cohort, and an IgG control cohort. You see an additional uptick in the tumor. These images were done with Cherenkov imaging, which is really nice because SPECT imaging with Lead-212 would take a lot of time, and with so many mice, it would not be feasible. So Cherenkov here is really your friend. You see the therapy impact. In our 30, 60, and 100 microcurie cohort, you see a delay in tumor growth. You see the quite quick progressing in the control cohort. So in the highest cohort, we basically have a median survival that is double as high as with the sodium chloride cohort. This is very nice and gives us first insight on how impactful Lead-212 is. But clearly that screams that multiple injections, so microdosing, would have been even more beneficial. And we want to do that as well. Before we jump into that, let's talk about potential toxicity in lead studies. And I mentioned before that bismuth, the first daughter, and actually the one that is doing the alpha trick, might escape. Does it really? If it would escape, that's a biodistribution of bismuth chloride in mice, it would majorly accumulate in the kidneys and would do kidney toxicity. We saw that in vitro studies, round about 35 to 40% of bismuth will escape the chelator, which is actually fantastic, meaning 60% is retained by the chelator upon decay from lead. That will not happen with actinium. All 3 daughters will be released from the chelator. But even though we see this release of bismuth from our chelator DOE3A, we do not see this amount of release in vivo. Actually, it's merely around 10 to 15% of difference that we see in the tumor. And the additional bismuth indeed is found in the kidneys. However, even though we used a really high dose, 100 microcurie, the toxicity that we found was minimal to mid-dramatic. We see here in the healthy kidneys, how a kidney should look like, that tubules are very nicely organized. In the treated mice, you don't see necrotic area, but what you see is healing kidneys. So the tubules are rebuilding and are not as organized. Surprisingly to us, not really surprisingly, but a little bit shocking, in the ovaries, that was the other organ that was affected. In the healthy tissue, you see here the eggs, the follicles, healthy and normal. And in our treated mice, these eggs were completely, there were no eggs anymore. So the mice were basically sterile. We do not really know where the toxicity here is coming from, if it's just the distance from the ovaries to kidney and tumor, or if there is really some slight radiotoxicity from the nuclide itself going on. We'll have to investigate that further. However, what we can say, due to the mild toxicity, we would like to do several injections of lead, so dose fractionation. This is a bit tricky for preclinical studies. The reason for that is if you buy a lead generator, as we have heard before, it's based on radium-224. And radium-224 decays quite rapidly over the time of, let's say, a week, 2 weeks. And so you elute less and less lead over the time. So if you want to have fractionated dosing and inject your mice every week or every second week, it's going to be really expensive buying these generators on a weekly basis. On the other hand, you can just buy the thorium source and make your own radium generators. However, this is quite tricky and you need to be quite the expert for that, and you don't want to touch that. So we thought about what is the stupidest, easiest way of doing that yourself without investing too much time. And as published before, you can actually use the radon itself that can escape basically the thorium source, capture it, and produce over polonium clean lead-212. We saw how easy, we wanted to see how easy we can do that. So what you see here is our own little lead generator. It's in a plastic bag. The reason for that is polyethylene actually is the best shield against radon. So everything that's in plastic is kind of safe. In the plastic bag, you see our lead brick, and in that one you have the generator. It's in a scintillation vial as an extra shield, and in that scintillation vial you can see the tube. And in the tube, the lead is glued on. You have the thorium in that little insert that you have. So actually that tube is pretty perfect. We have a drop of thorium in there, let it dry out, and just attach the tube on top of it. So what happens now is the radon emanates from that little spot and will accumulate in the tube and build up lead. You can see that in Cherenkov imaging, that's our tube. You see the radon basically streaming up and building up the lead here. And that's the tube from the top, where you can also see the lead is really in the inside, and we don't have a lot of contamination on the outside of the vial. And this is pretty perfect, because all I do now is I exchange the vial every day, add my buffer directly to the vial and the precursor, and radio label in the vial. So the effort is really minimal. There's also one downside, of course. The efficiency of the generator, since it's so simple, it's very low. It's around 20%. So if you're planning on doing that, you have to buy a bit more thorium. Safety is a big concern, so we're doing wipe tests. And if you want to start this, I would highly recommend actually having a scintillation counter aside and doing weekly wipe tests. With the plastic bag, we see that we actually have no contamination outside of the plastic bag. So it's a pretty safe approach. This all being said, thank you so much for your attention. I want to thank my boss, Jason Lewis, for letting me do all these stupid experiments, the DOE, and my collaborators for all of their support. That would be all from me.
Hey, thanks, David. We'd love to hear about those experiments. Okay, on to Jennifer. If you could share your screen, that'll be great. Thank you.
Okay. Hi, my name is Jen Bartles. I am a senior staff member in the research group of Dr. Susie Laffey, and I'm also a senior radiochemist in the UAB cyclotron facility. Just a little bit about what our facility does, so we are involved in the translational molecular imaging program at UAB, where we start with the production of radioisotopes, whether it be the traditional isotopes of fluorine or carbon, all the way up to radiometals. So we do isotope production, molecular imaging development, and then move into the in vitro and in vivo testing in small animals, and then hope to eventually end up into early phase and late phase clinical trials, and then start over again with our new radiometals that we've brought up. Currently, we have around 15 active INDs and several ANDAs in our facility. The UAB cyclotron facility, as well as the advanced imaging facilities, are located in the basement of the O'Neill Comprehensive Cancer Center. The AIF consists of 2 patient PET CTs seen over here, as well as a patient PET MR, and then there's a dedicated metabolite analysis space that is run by the cyclotron facility. When you enter into our cyclotron facility, you enter into our control room, which then is our equipment room and our cyclotron itself. In our production space, we have a dedicated QC lab, as well as a standalone radio pharmacy and a production suite, which houses 8 hot cells, as well as 8 different radiosynthesizers for fluorine and chemistry, fluorine and carbon. Additionally, we have a preclinical radiochemical space, where we do fluorine and C11 new compounds brought up there, as well as radiometal purification. Our cyclotron is a TR24 from ACSI. We can do variable energy protons between 17 and 24 MeV and can go up to 300 microamps on target. We have dual extraction port, allowing us to run beams on both sides at the same time at the same energy, and have 4 beam lines. At the end of our target stations, we have stations such as this that we can irradiate solid, liquid, and gas targets. And these pictures up here are an example of what a solid target station looks like, and I'll get into what the target looks like that goes into them. This is a 30-degree slant. On top of IND radiopharmaceuticals that we produce, we also do make a wide variety of radiometals. Most of the isotopes that are seen above are done by the students in Dr. Lappy's group, but we do also have routine isotopes that are shipped by the cyclotron facility, like copper and zirconium. Additionally, we provide 4 isotopes through the DOE University Isotope Network, including the one we're here to talk about today, which is Lead-203. So Lead-203 production is done or can be accomplished via 2 routes on proton irradiation of natural thallium. Thallium-203 has a lower natural abundance, would probably be a little bit more expensive to start with, but unfortunately, it also has a higher cross section to produce co-contaminants, including the undesired Lead-201. So starting with Lead-205 is a little bit better. We have a higher cross section and for Lead-203 production and a lower cross section for the co-contaminant. This is further shown here. On the left is the cross section of proton irradiation on natural thallium, and the green boxes are indicative of the cross section that we can capture. The best cross section, of course, would be a little bit higher, around 26, but we can still capture quite a bit. So on the left, you can see that I mentioned before, we would make a very reasonable amount of Lead-203, but also a very high amount of Lead-201. But starting from enriched Thallium-205 material, we would make more Lead-203 and very little Lead-201, and the Lead-204 would decay throughout the night. So moving on into production, I have to give a big thank you to Shefali Saini. She is our 4th-year graduate student who all of this work is done by, and most of the slides were provided by her. So a big thank you to her. And she's also probably on this call and could probably answer questions later. So for moving on into the production, Thallium-205 is unfortunately not provided in foil form. It is only provided in powder. Powders can be a little bit tricky for irradiation, so we thought the best path forward would be going to an electroplated target. So starting with a mix of the chemicals you see here, including the enriched thallium oxide, this is all dissolved at room temperature and about a pH of 13. This can be plated using up to about 30 milliamps and using either copper or gold. In about a 5-hour period, you can electroplate between 76 and 115 milligrams per square centimeter. And you can see that there's not too big of a difference between the surface. And I will explain why we have both copper and gold in a few slides. I would also like to point out that the results that are shown here, while you will see them on the slides, you can also read about them in one of the most recent journals of the Journal of Nuclear Medicine, which is where it was recently published. So moving on to the purification. So we start with the target dissolution, where the target is placed into a bath of 2 molar nitric acid and is gently heated to about 80 to 90 degrees C. It is then cooled to room temperature and then loaded onto the lead resin from iChrome at about 0.5 mil a minute. The thallium target material flows through to the waste and then is additionally washed with 10 more mils of 2 molar nitric acid, where it is then eluted in 5 to 6 mils of either 1 molar ammonium acetate or sodium acetate at pH 5. Originally, this is where our chemistry first stopped, and then in the early development stages, we sent some lead to some friends around the country, and they tested it, and we found out very quickly that lead is not stable in the ammonium or sodium acetate that it's shipped in. So that's when we introduced a secondary column, which would remove the acetate and provide it in a better form. So the ammonium acetate lead solution is then loaded, is washed with diluted hydrochloric acid, and then it gives our final product in a small amount, 400 microliters of 1 molar hydrochloric acid. This graph, this is just showing you the elution profile of the lead from the two columns. And you can see we lose very little activity within the initial loading as well as the washing, and then roughly 95+% of the activity is eluted in that ammonium or sodium acetate. Then after it's loaded on the second column, you can see it has a much higher affinity, very little breakthrough, affording our final product in a small volume, and we have average recovery yields of around 93%. To check the quality of the product fairly quickly, we do a titration using DO3 a chelator. We start with 10 mg per mil stock, and then we vary the concentration from 1 to 200 nanomole per vial, then incubated with approximately 100 microcuries of the lead chloride for 30 minutes at 37 degrees. And then we test the complexation of that using instant thin layer chromatography and EDTA as a mobile phase. And we have an average apparent molar activity of around 38 gigabecquerel per micromole. The final lead product was additionally tested for stable metal analysis using ICP-MS. And if you remember before, I said we could plate on both copper and gold. Originally, we started with copper because the copper is a little bit more inexpensive compared to gold, but you can see the breakthrough of cold copper in our final product was a little bit too high for our liking. Additionally, we also saw a little bit more thallium breakthrough, but once we switched to the gold, we significantly decreased our copper contamination, our cold copper in our final product, and our thallium levels are below limit of detection. One of the biggest things we've done this year is try to increase our production capabilities. So the coins that I showed you in the very beginning, those are coins that we can irradiate using our 90-degree targets. Due to the efficiency of cooling, on our water cooling on our cyclotron, we're only able to go to about 40 microamps of current, giving us production batches of around 3.7 gigabecquerels. However, if you remember the first or second slide, I talked about the 30-degree slant. This allows us to, it has better, the target itself has better cooling. So we were able to go upwards of 80 microamps on target, essentially doubling our batch sizes up to 7.4 gigabecquerels, and also increasing our apparent molar activity. So in summary, we have very nice electroplating methods with promising results, and this electroplating bath can be reused upwards of 10 times, just replenishing the thallium oxide between each electroplating. And then we're able to, bombard either at 40 or even all the way up to 80. We've gone a little bit higher, but 80 is about where we want to stop, using the 30-degree target design without any damage to the target material. We have a nice robust separation method between to get lead-203 in high purity from the thallium target material. And one of the biggest things is Shefali also transitioned this production down to the cyclotron facility team, myself included. And we're currently working on semi-automated separations, as well as ongoing work for recycling that thallium starting material. And this is just an image of a Zorenzo Phantom that Shefali captured this year using the UAB Small Animal Imaging Facility's new SPEC scanner. So with that, I have to give a big thank you to our team, including our director, Dr. Laffey, who's traveling right now, Shefali, and then our cyclotron team, JP and Jason. And of course, a big shout out to Perspective, Michael Schultz, who you'll be hearing from shortly. And we are a member of the DOE University Isotope Network. And as they mentioned earlier, Lead-203 or any other DOE isotope that we provide can be ordered through isotopes.gov. And with that, that's it.
Great, thank you very much, Jen. Look forward to some questions on that. While Kurt gets his slides ready, just like to remind everybody that at the bottom, you will see a Q&A tab. Please send your questions through the Q&A for the end for the moderated session. Thanks. Kurt, yours.
Great, thank you. I'm Kurt Zinn. I'm a professor at Michigan State University, and I'm going to talk about one of my student's work, a student who graduated this summer with his PhD, and he's now back in medical school doing rotations, and he's finishing up his MD, so he's not able to be giving this presentation today. We're interested in interventional based approaches for delivery of radioisotopes to tumors, and so that's what I'm going to talk about. We're interested in the bismuth-212 coming from this generator system. Currently, embolic spheres like TheraSpheres or the resin-based approaches with yttrium-90 are FDA approved, and they're kind of standard of care. This just shows you a C-arm fluoroscope where the interventional radiologist is positioning a catheter, through the leg, into the tumor that's in the liver, and you can actually see the catheter, through the C-arm fluoroscope. Then once he knows he's in the right spot, he can deliver technetium labeled MAA, or the actual therapeutic product directly to the tumor. And, basically this is what an image looks like after the delivery. So it's a very different approach than intravenous. You're going intra-arterial, trying to deliver directly to the tumor. What we were wondering, is there a way we could use an alpha emitter that might even be a better way than the current beta approved yttrium-90? So we decided to try labeling macroaggregated albumin with bismuth-212. MAA, as it's called, is a protein product. It's widely available, FDA approved. It's degradable. It lasts about 12 hours. So we thought we needed an alpha that matches the short half-life of MAA, and decided to use the bismuth-212 from this generator. Well, we're also motivated actually by the fact that we would like to be able to give fractionated therapy, and we think a shorter-lived alpha emitter would allow for that. So that's why we were kind of focused on bismuth-212. Part of this work has already been published earlier this year. I'm presenting part that's published, and then also extending some of our work more recently with multiple doses in tumors. So that's a little bit of background where we're coming from. Just to give you an overview, we're taking MAA, we're radio labeling it. We've done some work with treating cells in culture with escalating doses, and then looking at immune competent models to look at the therapeutic effect. So the radio labeling procedure is actually really easy. We buy the FDA kits. We're actually using them. We buy them from Cardinal Health. We incubate with the bismuth-212. And then we centrifuge, and whatever sticks to the particles, we rinse it off a couple times, and then do TLC. If it's not moving in our solvent that includes EDTA, it's good. If it's unbound, it's bad. The good stuff is at the bottom, and that's what it looks like. So this just shows you we've figured out a way to use an IVUS machine to image the TLC strips. It's kind of a poor man's way to have a TLC imager without actually having one. This just shows you free bismuth, not labeled, and then here's the MAA attached to the bismuth, and then we can look at the bottom of the strip here just to show that we've got the bismuth bound onto the MAA. So that's kind of our radio labeling procedure, very simple. Again, using an FDA approved product. So we grew up cancer cells in plates, and then did escalating doses. This is just one breast cancer cell line, the 4T1 cells with increasing doses. As the MAA particles, when they're added, they settle down, they coat the cells on the surface, and then they expose them to the radioactivity. So this is a significant treatment effects. We thought we needed to look in vivo to see whether or not there's any release of the radioactivity. So we actually injected tumors, and then did Cherenkov imaging. Then we can actually image the tumors themselves that are expressing luciferase, and then do the bioluminescence imaging with that. So the imaging shows that when we directly inject into the tumors that, we really do have the radioactivity there. Obviously, we can't thread a catheter down to the tumor. We have to, do what is best for the mouse, which is the direct tumor injection to mimic what it would be in an interventional procedure. If you follow that by bioluminescence imaging, you can see the two images kind of overlaid together, the Sarenkopf with the luciferase image. So what about other sites? As I mentioned, where could it go? We do that with measuring in a gamma counter, of course. In these studies, we do them at 2 and 4 hours, predominantly in the tumor. Almost 100% of the doses in the tumor, a little bit in the blood and the bone. And this is for one model that we have, the 4T1 model in the BALB-C. So here's the results for the plan for the therapy studies. Again, treating with the bismuth 212 labeled MAA at day 7 after tumor growth, and then measuring tumor size. And this is what we find, with the dose escalation, 25 and 50 microcuries here, a significant reduction in tumor growth by the treatment. You might say, what happens if you give multiple doses? And so this is a new data where Nate is actually treated 3 times with 25 or 3 times with 50. Again, fractionated doses. This is the result from that study. And here's what the tumors look like from that study. In terms of toxicity, we haven't seen any effect on the animal weight. We've collected tissues and had a pathologist look at them and found no lesions in any of the tissues that we've looked at. We're particularly interested in the lung because particles could be trapped there, and no toxicity. We've looked at red blood cells, platelets, and white cells, and have seen no effects from that perspective. So no toxicity so far. So just in conclusion, we think this is the first use of bismuth 212 for what would be an interventional procedure. Direct injection of the labeled MAA remains in the tumor, confers a therapeutic response, could be applied to various types of cancer beyond just what we've demonstrated with breast cancer. I showed you today just one model. Our publication has 2 different breast models, and we've extended that to now to melanoma as well. The fractionated therapy is safe and efficacious, and no toxicity was observed. So, what we're really excited about for the future for these studies is because these are immune competent models, we hope to, study this radiation therapy in combination with checkpoint inhibitors to achieve a systemic immune response. That's really our goal, for our future work. And with that, I want to again acknowledge Nate Kaufman, who was a student on the left here in the picture, who did the studies for his PhD work, part of his PhD work, and the rest of the members of the lab that supported him in that effort, and acknowledge the funding source for this work. So thank you very much for your attention. I appreciate it.
Thanks for sharing that, Kurt. Very nice to see some more applications and a different approach, especially the bismuth 213 side of things. Thanks. Mike, you have some slides you can share for us? See you. Thanks.
Mike, you're still muted.
Thank you, David.
Thanks, David.
First of all, I'd like to thank the organizers for the invitation to come and speak today. I'm gonna sort of riff off several of the points that have been brought up so far today, and I think add some more exciting information about some clinical successes of this idea of lead-203, lead-212 alpha particle radionuclide therapy. These are my disclaimers. I think the exciting thing today is that, you know, radiopharmaceuticals and image-guided radionuclide therapy for cancer are really emerging as a pillar in oncology. And there's lots of exciting parts to this slide where we heard just recently about, you know, the potential for combination therapies. The imaging, of course, gives us this, you know, imaging surrogate that can help us to do treatment planning early. And certainly we're seeing from the preclinical experiments that have been presented so far today that the, you know, the imaging is really an important aspect for, you know, de-risking compounds and developing compounds as we go. And there certainly are, you know, a number of targets, you know, many targets left for us to explore in this field. And you know, this sort of bears out in what we're seeing in published literature today about whether you know, however you spell it, theranostics or theragnostics, are certainly emerging as an important aspect. And this is just a PubMed search that I did on these two search terms. And you can see this, you know, steady increase in publications that are, you know, that are really focused on the kinds of things that we saw today and expanding the applications for these beyond, you know, beyond where they started. And so it's a really exciting time to be in this field. And you know, it's not just us radiopharmaceutical scientists who are starting to recognize the potential for this. You know, this was, you know, the ASTRO meeting now is, you know, is beginning to fill with, you know, radiopharmaceuticals. And one of the things that's really emerging here over the past several years is that this idea of alpha particle radionuclide therapy for cancer really has the potential to be, you know, transformative for cancer patients. And it's increasingly recognized that that's the case. And that's really the reason why all of us are here today, to explore this idea of what is it about Lead-212 and Lead-203 that lend themselves so nicely to the type of radiopharmaceutical therapies that are emerging in, you know, over the last, you know, half a decade or so. And I think, you know, a few things have sort of emerged that really are important for the Lead-203, Lead-212 story. One, peptides have emerged as, and peptides and peptide-like small molecules have emerged as, you know, the targeting ligands that have the best potential for, as Dr. Bauer brought up, you know, delivering the radiation specifically to the tumor microenvironment, you know, fast PK properties, residual dose can be washed away. So with that, it just really makes sense with the kind of PK properties that, you know, that peptides have. You know, here's an example of, you know, antibodies versus peptides. And, you know, there's a lot on this slide, but I think the key takeaway message in the kind of time that we have here today is this idea of, you know, fast biological kinetics, high tumor penetration, rapid clearance that can be largely, you know, engineered to be renal and excretion, short biological half-life. And those kinds of properties really lend themselves to Lead-212. And so if you think about sort of the natural history of theranostics in general, we've had iodine-131 for many decades as an effective theranostic isotope for thyroid cancer. Later on, we started looking at peptides and antibodies, and we figured out that we could attach, you know, beta particle emitters like lutetium-177 and yttrium-90 to those molecules very effectively and this whole idea of, you know, being able to target the tumor microenvironment more effectively. And then, but really over the last, you know, 5 or 8 years, we've really recognized that alpha particle radionuclide therapy could be transformative for cancer patients. And Lead-212 with a relatively short half-life, as Dr. Bauer brought up, really lends itself to this peptide targeting. And there's, you know, there's other aspects to this that have to do with, you know, supply chain and cost of production that also lend themselves to the Lead-212 story. But just purely from a biological perspective, it really made more sense to me as a developer of radiopharmaceuticals to use an isotope that matched up better with the PK properties of the molecules that we were intending to use. The other part about, lead-212, if you're new to this, and you're just investigating lead-212 and lead-203 for the first time, is the potency of these alpha emitters in comparison to beta particle emitters. So this is a study that we did. This was presented at the EANM meeting last year by one of our scientists. And here what you see is, an AR42J model that's very aggressive. We treat it with a fractionated approach that is, comparable to what's done in the clinic with lutetium-177 dotatate. But then when we compare that to either a single dose of our lead-212 labeled VMT AlphaNet versus 4 fractionated doses, you can the potency of lead-212 in comparison to beta emitters becomes, remarkably clear. And so it's potent. It matches up with the PK properties of these peptides and peptide-like small molecules. But also, we have this elementally matched isotope in lead-203 that can be used as an imaging surrogate. And this turns out to be extremely valuable, not only in the preclinical setting, as Dr. Bauer said, talked to us about, but also in the clinical setting for, in oncology, this is a rarity to be able to, quantitatively make determinations about the potential effectiveness of your oncology therapy for cancer patients. That's a really powerful tool. And because the lead-203 and lead-212 are elementally identical, we can trust that the biochemistries of the molecule will be identical as well. That makes the lead-203 an excellent tool in making predictions. Now, we've talked a little bit about chelators today, and we developed our own chelator early on in this, and that was, primarily because the chelators that were available, I felt weren't really engineered specifically for lead isotopes. To a first approximation, one of the things that we noticed was that they leave a net formal charge at the radiometal chelator coupling. And we know that the kidneys are particularly, the proximal tubules in the kidneys are particularly adept at taking up charged species. So by reducing the formal charge to 0, we expected that we would be able to improve on renal clearance, and also from chemistry first principles, improve on radio labeling and stability. And it turns out that, the chemical modifications that we made, seemingly small to the chelation moiety of a somatostatin targeted peptide, such as triatides. Here you see an example of how you can make, relatively small changes that reflect, significant improvements in tumor targeting and other organ, and renal clearance. So here you see an example of, lead-203 DOTA-TOC, which is, as many of you know, FDA approved radiopharmaceutical when labeled with gallium-68. There's roughly a 1-to-1 ratio between tumor and kidney, but by making some small, what seem to be small molecular changes to the molecule, we make a significant improvement in tumor targeting versus renal retention. And of course, the important part there is that those types of properties are also imparted on the molecule. When we get into the clinical setting, and here you can see one of our first patient studies. This was done by Dr. Vikas Prasad and colleague Dirk Mueller, was published in Clinical Nuclear Medicine in 2022, where we recapitulate this very fast tumor targeting, rapid renal clearance, even seeing liver metastases by SPECT-CT on this whole body image at 1 hour post-administration. But then also, just as important, or maybe more important, we see this high retention of the agent using this lead-203 version of VMT AlphaNet in the clinical setting. So really nice tumor targeting that is, recapitulating what we had seen in the preclinical setting. And of course, recently published or presented at the European Association of Nuclear Medicine in Vienna and Austria by our colleagues from Fortis Hospital, Dr. Dharmender Malik and Ishita Sen, presented on some of their first work with this lead-212 alpha particle radionuclide therapy for neuroendocrine tumors using the VMT AlphaNet molecule. And here you can see, this is on the upper left-hand corner here, you see the tumor before the initiation of treatment, about 60 days before the initiation of treatment. Just after a single dose here, you see, a significant improvement, not only in, the tumor targeting by the PET-CT here using gallium-68 dodonok, but also, an appearance of resolution of the tumor by the CT. And then, after 3 doses, clinical follow-up imaging shows not only is, the tumor appear to be, nearly completely resolved, both by imaging, by PET-CT imaging, as well as by, CT, but also this patient has had a significant improvement in ACTH levels, showing that, they're feeling better, able to, go back to work and pursue professional activity. So a real exciting demonstration of the power of this, not only in the preclinical setting, but also in the clinical setting. In terms of what, other indications that we're pursuing at Perspective Therapeutics in this Lead-203, Lead-212 story. Here we show some images that were presented by Dr. Jeff Johnson at Mayo Clinic in the at the Society of Nuclear Medicine meeting earlier this year in the United States. We're targeting the melanocortin subtype 1 receptor, in this case for metastatic melanoma. We have gotten significant support from the National Cancer Institute, including some work that we're doing in combination therapy. And here what you see is this concordance between FDG and a Lead-203 and a gallium-68 version of this MC1R targeted peptide, along with the Lead-203 imaging, which is helping us to be able to move this agent forward in a clinical trial that is open in the United States now. So based on this idea of Lead-203, Lead-212 image-guided radionuclide therapy for cancer, and and the proprietary chelator that I talked about, as well as a deep bench in our discovery lab, we've developed a nice discovery pipeline of proprietary molecules that are advancing at different stages of discovery and first in human therapy and phase 1 trials. And we're excited about the potential for that. And one of the other things that comes up about Lead-212 is the idea of the manufacturing and distribution of the finished product radiopharmaceuticals. And so this slide just kind of gives you an idea of how we're building on through collaborations with existing networks of radiopharmaceutical manufacturing facilities in North America to develop a series of sites. Here you see 6 or 7 sites that we're expanding into over the next year and a half or so to be able to build out our clinical trials. But then we're also taking advantage of the over 100 sites in the United States that are recognizing the potential of these therapeutic radiopharmaceuticals and expanding their facilities to help us to be able to deliver these from these individual sites around the country. So that's a little bit of highlights for what's going on with Perspective Therapeutics. We're excited about the Lead-203, Lead-212 story. We are, really focused on 2 clinical stage programs right now that are in first in humans clinical trials. And we believe that this Lead-203, Lead-212 dual isotope theranostic approach just has real potential to be transformative for cancer patients. And so with that, I guess I'll stop there. I'd just like to thank everyone for tuning in today. We always like to thank the National Institutes of Health and the National Cancer Institute for all the support over the last several years, and the Department of Energy and the NIDC for their support in getting the Lead-212 and Lead-203 story off the ground for cancer patients. And I'll stop there.
Great, thanks, Mike. Very nice to see some more in-human data. Big thank you to all of the panelists for taking the time to prepare slides and share their exciting work with us today. Let's now move into the moderated Q&A session. David, I have 2 questions for you. The first one, so they both come from Lake Witten, from Morona Med. He asks, are you willing to explain a little more about the release or capture of daughter radionuclides after the initial decay of lead-212?
Yes, of course. That is an excellent question. So when you follow up the decay chain, you will see that lead undergoes electron capture, and therefore bismuth itself would not really be ejected from the chelator. There's a really low energy, and so we can assume that a chelator, and DOE and TCM are both kind of good chelators for bismuth as well, would remain in the chelator and not being released. However, there's one more effect happening, and this is internal conversion. And so some bismuth species might end up in like 7+ stages, and this would just strip the electron from the chelator and destroy the chelator, and the nuclide would be released. So we know that this internal conversion only happens to like 37% per decay. So which means that for sure 60% of the bismuth, and we find that in the studies, are retained in the chelator. How could you study that? Well, I hope really that my publication will be out in the next few days, 2 weeks, and then you can read that up, how I did that. However, there's already so many publications in the literature. You can just do a normal cartridge, capture your compound on the resin, and then just elute basically 3 daughters and measure them in the gamma counter. You have an H+C method that you can use. You can use antibodies and spin filters. You can even base it on an ITLC, even though it's a little bit trickier because you really want specifically go for the daughters. So you would have to still go gamma counting. But there are many ways you can read it up in the literature, and you should really do that if you start a study, just to see how good is your chelator.
Thanks. And here another question. It's related, I guess, to you mentioned about doing swipe tests. So could you share some more details on method for your liquid scintillation of swipes? What kind of swipes? Using a cocktail, counting settings, that kind of thing.
I see. So the swipe tests are pretty straightforward and standard everywhere. The only thing that you need is the wipes, or you can literally just use Q-tips. You wipe an area of approximately 100 square centimeters with this, and then you put your wipe test, your Q-tip, in a scintillation vial and fill it up with 3 milliliters, 5 milliliters, depending on the tube, of your cocktail. We use Ecosint A, I think it's called. And for the scintillation counter, we use a Perkin-Elmer one, but everyone is fine. The settings, however, are quite interesting because you can differentiate between energies. So you can differentiate between low gammas, electrons, and alphas if you have the right settings. You can find that in the literature. And we go only for the alphas. Main reason is if you work in the labs with many nuclides, you will always have slight contaminations, and you want very specifically only go for the alpha emissions. So have the right high energy settings for alphas, and then with your wipe tests, you will be all fine.
Thank you. I had 2 questions myself for you. You showed a biodistribution done with bismuth-213 chloride, and I was curious, the source of that bismuth, was that from a generator produced? And then, you know, if you're doing that, what are your elution conditions? Or is that from your thorium-228 generator from emanation? And then how are you extracting that?
Very good question. So this was actually from a DOE derived generator.
Okay.
We did the dual approach to purify lead. If you want to elute bismuth, you just go with a lower concentration HCl first. So you mainly elute bismuth, and most of the lead will be staying on the generator. After that, we still put the solution through lead resin to just purify out all possible traces of lead-212. And this repurified bismuth we injected into mice.
Thank you. My last question for you was to do with your emanations work. Very exciting. I like to see that. You mentioned about a 20% yield. Is that 20% largely due to radon escaping the Ziploc, or is it just low extraction because of the solvents that you're using?
Very important question. Yes. So partially when you have the emission of radon polonium to lead, this alpha and recoil can go both ways. So a little bit of lead will be lost in the plastic. However, that's only a small fraction. And it's not the radon escaping the bag, but it's mainly the radon not making it out of the lid. The half-life is 1 minute. That is quite short, but radon is, for a noble gas, extremely sticky. So with this super simple setup without any gas exchange, you might just not be able to get most of the radon into your tube.
Okay, so staying in the lid. Okay, thank you. I have a question here. Not sure as Johnny asked the question, great to see in vivo preclinical and clinical systemic use of lead-212, a lot of attention paid to kidneys. What about bone marrow effects, blood counts, toxicities, early and late after dosing? Not sure who he's directed to. Yeah,
I can try and help it out. That's a great point, right? So I think that kidneys often are, people pay attention to kidneys because the clearance route for these peptides and peptide-like small molecules is generally, through the renal system. And so it winds up being an important, potentially dose-limiting organ, although we find that oftentimes, particularly with the kind of clearance properties that our peptides wind up having, that the bone marrow can wind up being looking like the dose-limiting organ. In our preclinical work, what we found in comparison to other isotopes like actinium, for example, that we get a lower toxicity readout for all of those important parameters that you're talking about, and in this case, particularly in the kidneys, and that primarily, we believe, is due to the escape of bismuth-213 from the chelator on the decay of actinium-225. And by the way, for a nice publication on that chelator stability, we published, it's in my presentation, but Lee et al. In Pharmaceutics in 2023 is a nice demonstration of the stability, actually the stability of bismuth in the chelator in formulation. So there's some differences in some of the experiments that Dr. Bauer has done, elegant studies. But we find in formulation that the bismuth-212 on the decay of lead-212 is pretty stable. So we'd invite people to take a look at the publication that we have on that.
Great, thanks, Mike. While you're up on my screen, at least, I have a question for you. I understand, at perspective, you have to match the biological half-life of your vector with the rheological half-life. That's an important thing we've talked about that today. It's come up. Also, it's of interest with the theranostic pair, you have that. But are you within your pipeline investigating potentially use of bismuth-212, or really just focusing on lead-212 based drugs, radiopharmaceuticals?
Well, I certainly was really interested in Dr. Zinn's presentation today. That was a really elegant way to be able to try, the whole point here is trying to deliver radiation to the tumor microenvironment and limit, the potential for off-target toxicities. And for this indication that he was working on, it looks like he's really got something there that's exciting. However, with a 1 hour half-life, bismuth-212 as a finished product radiopharmaceutical probably isn't feasible except for manufacturing on site. So you could deliver for the MAA application, you could deliver lead-212 to sites for them to be able to extract the bismuth, maybe by a radium-224 generator. But that'll be a challenge for widespread deployment of finished product radiopharmaceuticals, where lead-212, with a 10.5 hour half-life, is can readily be deployed through a local regional manufacturing and distribution network, like I showed.
Sure. You raise a good point, I guess. Coming up with 110-minute half-life of F-18 always on site, right?
Well, actually, that's a really good point. And when you think about that network that that we're talking about taking advantage of, fluorine-18 has a 2 hour half-life, and there's a national network of manufacturing sites, through several different, CDMO type radiopharmacy operations, right? And we all know what, who those groups are. And with a 10.5 hour half-life, lead-212 can easily fit within the logistics that already exists for manufacturing and distribution of lead-212 compounds. So great point. And those organizations, and we have publish things on our website about the different organizations that we're working with to be able to, to make that network happen for this Lead-212 radiopharmaceutical therapies.
Great, thank you.
Great question.
I have a question for you, Jennifer. I was looking at your nuclear reaction slide, and you plotted some cross sections, and it looked like that the cross sections for enriched targets were higher. So I was curious about how does the cross section for Lead-201 look for the enriched material at lower energies? That data was really missing from the graph, and I was curious to see if there was some kind of similar trend with the local maxima at the energies you're irradiating at.
I can't answer that completely, but I can tell you about what we see in our final product.
Yeah.
So Shefali has done some calculations, and she's determined that we can make it max, probably end of bombardment, maybe 2%, but we're generally way less than that. So final product, we have moved our bombardments to Mondays, and then we decay through the night and produce or do the production on Tuesday mornings. And we are averaging around 0.3% of Lead-201, which seems to be okay with customers. But if we move our bombardments back to Fridays and then produce on Mondays, which is what we were originally doing, you don't see any Lead-201. But we wanted to maximize our production. And so since we're below most radionuclides that are, FDA regulated, you want, 99.5% radionuclidic purity. So since we're below or above that purity, we're pretty okay with that. I'm not sure if we're going to do cross-sectional measurements just to determine how much we would make, but that's at least what we see. So.
Okay, thank you. Separation that you showed, I wouldn't say it looks trivial, but it looks quite simple. What is the total processing or separation time with a 2-column system?
Time-wise, it's about an hour, honestly. So the dissolution of the thallium from the target backing is around 5 to 10 minutes or so. And then at the flow rate and the volumes that we use, I mean, it's really from collection of target to final product in hand is a little over an hour. We have done some testing with some semi-automated, like using valves just in place, like not in a synthesizer yet. And that will reduce the time. We've got it down to about 45 minutes, but we've lost a little bit more in the lines. But we're still working on that, and hopefully by maybe next year we'll have something better in place, and hopefully in a manuscript form. So yep.
Oh, great. Yeah, fast isn't always better, but
right,
always helps, right?
Yeah.
I had another question. So you mentioned about the radioisotopic impurity of the 201. I guess, are there any other radionuclides that are produced or activated or slipping through the, that customers can be aware of in the final product?
We haven't seen any. If we were, I learned the hard way one day doing a production on the same day of bombardment, and there is a lot of Lead-204. But it's got a very short half-life, so it decays away through, through shipment, of course. And beyond that, it's just that Lead-203. I haven't seen anything. The only thing cold, of course, was the copper. But radioactivity-wise, we don't see you would activate the gold. Of course, but we don't see the mercury isotope in the final product. So we're pretty happy with that. But yeah, it's mainly just Lead-203.
Okay, thank you. I see Matt has his hand up. Do you have a question for Jennifer?
Yes, I do have a question for Jennifer. So I was curious, so with your lead resin purification process, you mentioned issues with stability of the lead product in the ammonium acetate. And I was wondering if you could expand on that a little bit.
Sure. So I don't know a lot about it, but I can tell you what I do know. So when we were collecting we were doing fraction collecting in about 1 mil fractions in the either ammonium or sodium acetate, and we did ship it to a few academic sites just to see if they could radio label. We always managed to have decent radio labeling the day of production, but then when we would hear back from our friends at the sites the next day, they always had issues. And I don't know if it was the concentration, because it was maybe a max of 1 mil, whereas now we're shipping total in 400 microliters. I could probably ask Shafali or see, but it was other sites. It wasn't necessarily us. So yeah, it maybe not stability, just like inability to label, but it could be conditions. It could be a variety of things. Yeah.
Okay, and so did you see the, you mentioned sodium acetate. Did you have the same issue with both salt forms?
That honestly would be a question for my colleague. I know we currently elute in sodium acetate, but it doesn't, I don't think there's really a difference eluting between sodium and ammonium, to my knowledge, but I could be wrong.
Thanks for that question, Matt. Matt, I had a question for you. I guess I'd love to do a head-to-head comparison on the generators that you will start producing in the winter to the generators that we're producing. Something I was curious about was what is the column volume of resin that you're using or will use?
So we modeled the generator after the Oak Ridge generator.
Okay.
So it's effectively the same thing with the actual bed of the column that contains the homogeneously distributed radium is right at 260 microliters with a 50 microliter bed of non-radium bearing a catch bed below that. So I think that's very close to what Oak Ridge is.
Yes. Yeah, above and below that kind of volume, correct. And then you mentioned doing some thorium-228 breakthrough in your separation. Could you comment on what kind of percentage breakthrough you've seen in some DK studies? Thorium-228 present in your radium-224?
So not measurable. You saw that it was basically I reported 2 to the -8 parts thorium on the generator itself from the original thorium piece.
Okay.
So 300 picocuries on the generator, and the thorium is bound much stronger than the radium itself. So any thorium that ends up on the cation exchange resin has a very low probability of ever breaking relative to the radium breakthrough.
Okay.
I think the radium distribution coefficient on the resin is what, 2 orders of magnitude lower than that of thorium, I believe. So yeah, it's not going anywhere.
Thank you for coming in on that. So I have a question in the Q&A box. Looks like it's for everybody, all the panelists, please. Thierry Storer from CERN Medicis, Switzerland says, thanks, Andrew colleagues, DOE. They're starting up with their Lead-203, Lead-212. Is anyone concerned about the 7 to 10% approximately may have high energy gamma branching for clinical translation or handling? Mike?
So great question. And yeah, absolutely. Just like, for any other, radioactive materials, you want to look at what the decay properties are and how they need to be shielded. And there's certainly a concern. I think that that for, the clinical operations, when we look at the likely doses that we're going to see, administered radioactivity for patients, and we've modeled the scenarios, we don't see that there are radiation safety protocols outside basically of what the norm is for, the nuclear medicine clinic. For larger sources at the production sites, you'll have to have appropriate shielding. But I think from the clinical scenario, it doesn't look like it's going to, be a big change for the nuclear medicine technologists and the other, personnel that are working in that area. but you do have to watch for that and shield it appropriately.
Definitely with higher activities, significant shielding is going to be required at production facilities. Thank you, Mike. Veronica Mako asks if you could describe a little bit how we load the generators with radium-224. So she's curious to know if we load it in column format or do we do it in batch. So I'll talk a little bit about that, how we do it at Oak Ridge. And she wants to know how we assay the amount of radium-224. So the separation that we're doing from our thorium-228, we do a 2-column separation to first remove the radium from the thorium. And once we have that radium solution, we remove the daughters, really just to, one, as a help for a cleanup, and then I guess to remove the dose from that particular radioisotope, so our operators who are loading it receive less extremity dose and body dose. So the final solution, we assay that and we determine the amount of radium-224. So since we've stripped out the lead-212, it's really easy to see the gamma emission of the radium-224 itself. Once that activity concentration has been quantified, then based on the work authorization that we've received from the NIDC for the customer, we can then dispense that amount of activity of radium-224 into a vial, which we contact with the resin. And as Matt and I were talking earlier, and we saw that in the image that I showed with Sharenkov, you really see the non-contacted resin. So we put a small bed, which would essentially act as a catchment for any radium that breakthrough on the generator. So you shouldn't see radium in your lead-212 or bismuth-212 elutions of the generator. And then the radium-224 is loaded onto that. It's contacted with the resin, and then it's homogeneously distributed over that resin. That resin then is loaded into the generator body itself, and then a portion of non-contacted resin is loaded on top, sealed up, and then we do a final stripping of the lead-212 for packaging. So really when it's going out, we have to take into consideration the ingrowth of daughters towards the TI and for shipping. We have a multiplier for that to make sure that the correct TI is noticed at the customer's receipt site. We do not do a milking to determine the amount of lead-212 to then decay correct for radium-224 generate assay that we've loaded. We do look at the dose rate on that vial to determine that all of the activity has been loaded. Hope that answers your question, Veronica.
And I'll just step in. Basically at P&L, we're doing essentially the same thing as what Andrew just explained, except it's mechanized, and the radium, yes, is homogeneously distributed throughout the bed of the resin.
So I have a question from an anonymous attendee for Matt. They ask, what dose rates are you seeing for operators manufacturing your approximately 30 millicurie generator in each type of manufacturing setting, whether it be in the hot cell or a glove box? Do you have an idea of that value?
Is this question, is this getting at personnel dose? If so, I don't know. I don't know if I can answer that.
Oh yeah.
It's how about I say it's very modest.
Yeah, I think that's good value. You know, if I think about the operations here at Oak Ridge, since we're only working inside of a glove box, you know, our cow, which is typically in the range of 20 to 25 millicuries at any one point, probably on contact reads around 6 R per hour. And we use appropriate shielding where we can to not hold that as close to oneself as at any time. And as I mentioned, we strip out the lead as quickly as we can to reduce that dose. And operator dose within 1 calendar year is within our operating license. We're not exceeding that or asking for extensions in any case. And could be milking our cow in the order of frequency-wise every 3 weeks.
Yeah, and for us, basically, you know, our extremity dose is extremely low because we are not going in and handling that feedstock solution in any way, shape, or form.
Yeah.
out the backside of the generator when we have to physically go in and disconnect it and get it out of the glove enclosure for packaging. As you mentioned, Andrew, the last thing you do in the generator is to strip any residual grown-in lead. And so that drops the dose way down. So we're in the so that generator, when it comes out, is in the millirem range. So very low contact dose on that. It's a completely different story when the customer goes to open the pig on, in 24 hours. Now you're in the R range dose on that same device.
Kurt, I had a question for you. I guess I'm not too familiar with the radio labeling of MAA. I was curious, is that a direct labeling of the bismuth 212 directly onto it? So there's no chelator involved?
No chelator. It's a direct labeling process. We do it in about 10 minutes at 70 degrees, and it's MAA directly that we weigh out from a FDA approved kit. Those kits have a predetermined amount of tin chloride. There's just enough apparently to reduce the bismuth and bind it tightly onto the particles. I mean, we've tested it over 4 hours, of course, and it doesn't come off. I mean, in serum or in other conditions, so pretty tightly bound on there, but a very easy approach. Hopefully that'll make it go into a clinical trial fairly easily, because it's already an FDA approved product and it's just a different isotope.
And then does that mean, you say, you talk about the ease of it, no purification needed for this ready labeled MAA?
Well, the purification that we do is that we spin the MAA particles down and supernate and then wash it. So we do a step to remove, I would say 70–80% of it sticks to the particles. We haven't tried to optimize that part because it's so easy to, to clean up by centrifugation.
Oh yeah, thank you for explaining that. I have another question in the Q&A box. It's for Mike from Jinder Fan. She asked, for clinical studies, were the Radium-224 generators complied with CGMP regulation, different from preclinical generations? She's asking about clean room, things like that.
Right. So important thing there. So those studies were not our studies. So that was under compassionate use under the direction of the physician at the Fortis Hospital in India, and done under regulatory standards that were acceptable under their IRB and under their statutes.
Great, thank you. Well, seems there are no further questions. We're a little bit early, we're a little shy of the time, but doesn't mean we can't adjourn. So we'll adjourn today's 2013 Department of Energy Isotope Program virtual seminar series on Lead-203 and Lead-212. I'd like to thank all of the panelists and the attendees, everyone who's participated and asked questions. I hope you have a good rest of your afternoon, and we'll see you next year. Thanks and goodbye.
Thank you.
Thank
Thanks,
you,
everybody.
Andrew and Jacob. Take care.